Marcio, thank you very much. Thank you for the introduction. Thank you for the invitation. And congratulations for this very well organized and large event. And it's a real pleasure to be here again in this conference to talk to you a little bit about our knowledge on the development of the intestinal microbiota of foals. So first, we should understand a little bit why everybody's giving so much importance again to the microbiome or to the microbiota. And it's important to say when we refer to microbiota, we're going to be talking about mainly bacteria, why they are so important. As you know, in the past, we used to use culturing methods to study and to investigate bacteria. But what the scientists already knew at that time is that when you look under the microscope, only between 5 to 10% of these bacteria can actually grow on those traditional culturing media. And therefore, the large majority of the bacteria remain unknown until recently, when we started to use DNA sequencing to investigate the microbiome. That means those bacteria, they might not grow in the traditional culturing media, but they are still there. And now we can go and just have a big picture of everybody through the DNA that is present in the environment. And over the last decades, those technologies became much smaller, easier to use, much cheaper, which allowed us to have a much better idea about the importance of the bacteria in our lives. Just to give an idea, if you type microbiota in the PubMed, what you're going to see is a huge increase in the number of publications in the last few years. And that started when Roche released into the market the first compact, small DNA sequencer that everybody could have in their lab. So that gave us an average of 50 new publications per day, just indexed in PubMed, which is kind of crazy to keep updated in this field. And revealed us that we are living in a world that is taken by bacteria, right? They have been here much longer than us. If we think that when we compare our genome with a corn, we are much more similar to a corn than when we compare any two species of bacteria, telling us that they are much more adapted, they are much more diverse and have been living here in this world. And we actually evolved in a world already full of bacteria. And that's the reason why we carry so many of them on us and inside of us. If we stop to think that we have one human cell to each bacteria in our body, that allows us to say that we are 50% bacteria and 50% humans. So they gotta be important somehow, and that's why so many different projects are studying the microbiome of humans, and they are designated millions of dollars to those projects. But us in the equine world, we work with a much smaller budget, but we are starting to have some ideas and clues about the importance of the microbiome to horses. So the objective here today is first to give you a little idea about what we know now about the microbiome and the development of the microbiota of foals. And then, just because there is not a huge amount of data up there, I also want to discuss a little bit of the future perspectives, or at least of my point of view of what we're going to be using that data we have now in the future to treat and to improve performance in horses. So why is the microbiota so important? Probably Dr. Mariano will touch a little bit about this in the next lecture, but we know that germ-free animals, they don't fully develop their microbiota, the intestinal mucosa, if they don't have bacteria there, okay? So the bacteria is somehow helping and developing the structure of the intestinal mucosa, which is also have a huge impact on the absorption capacity of that organ. After that, we also know that the microbiome is important for the immune regulation of the local and the systemic immune system. For example, germ-free animals, they have a completely dysregulated immune response, because this bacteria, when it gets in touch with the immune cells in the intestine, it is very important to not culminate in a very hyper reaction later in life. And we're going to talk a little bit more about this in the next slides. Finally, and also the protection against pathogens. So the intestinal environment, it's a place that all the bacteria wants to be there, because, you know, it's a perfect environment for them. It's free food coming all the time. It's no oxygen for the anaerobic bacteria. So they all wanted to be there. And therefore, there is a huge competition. We have the classical example of treatment with antibiotics. When humans or horses, they get a course of antibiotics, what happens is those drugs will just vanish all the, or most of the good bacteria, and that's going to open a huge door for the pathogenic bacteria to cause disease. There's a classical example in humans with treatment of antibiotics that develop Clostridioides difficile infection, mainly because, you know, the good bacteria are gone, so now the pathogens can grow and produce their toxins. Also in lab animals, the infection dose of Salmonella drops tremendously after we treat them with antibiotics. So that proves that this good or beneficial commensal microbiome, it's very important to protect us against enteropathogens. Now, I don't have to tell you, because maybe you probably know that better than me, that the intestinal bacteria, of course, they are very important also in the nutrition point of view, because they are responsible to produce energy by the fermentation of structural carbohydrates such as celluloses. So if you have, there are many studies showing that germ-free animals, they will gain 30% less weight compared to controls eating the same food, but just because they don't have their microbiota or their bacteria, they gain less weight. And this is attributed to the production of volatile fatty acids that are the byproducts of fermentation that can be absorbed and used as energy source. So we know, especially in horses, that the intestinal microbiota is very important to getting energy or extra energy further than the animal's digestion into their bodies. Okay, so let's see what we know about the microbiome of foals, healthy foals and sick foals. But before we start, I just would like to teach you or to show you one type of analysis we normally do to evaluate the microbiota. This is called principal coordinate analysis, and it's basically a graphical representation of similarity between the microbiome of two different samples. So when you have two dots, one beside the other, it means that those two animals, they have a very similar microbiota. But if the dots are far apart from each other, it means that their bacteria is completely different. Okay, so it's very simple, but we're going to be using that a lot. And as you can see here, we can see two different clusters. One, this is a study we did here, giving antibiotics, so sulfatrimethoprim to horses. And we can see that after 5 days of treatment with antibiotics, that microbiota changed completely. Those are the same animals, but it's just a completely different composition before and after antibiotics. So I think this is quite clear to understand. Just to give you another example of this type of analysis, in this study, they evaluated horses adult horses in 3 different diets. So you have hay, pasture, and silage. And what you can see is that depending on the diet they get, they will have a completely different microbiota or completely different bacterial composition in their feces. So that study also shows that diet is an important factor in the composition of the microbiome. Good. So coming back to foals, what we can see is that, what we know is that the first bacteria that will colonize animals are the ones in the birth canal, okay? For example, in this study that they show in humans, they were comparing here, as you can see in green, the oral microbiota of woman mothers. And here, the microbiome of human newborns that were delivered by C-section or natural birth. And what this study shows is that babies born by C-section, they were colonized by the skin microbiota of their mother and the nurses, while babies born by normal delivery, they had a microbiota similar to the vaginal microbiota of their mothers. So the first bacteria colonizing animals and babies are the ones present in the birth canal. For example, in that same study, here's the composition of the first fecal microbiota of the baby one, which is identical to the vaginal microbiota of his mother. And again, if you look at the composition of the microbiota of baby 3, it's just very similar to the microbiota of his or her mother. And again, with baby 4 is the same thing. So the composition of the vaginal microbiome will start with the colonization and shaping the microbiome of their babies. This was a very nice study done in horses that I had the pleasure to review. And what it shows to us is, again, the composition of the mare microbiota. We have here very clearly distinct microbiomes in the mouth of the mares, in the vagina, and in the feces. So you can see that there is a clear differentiation between all the environments. And what was interesting is that the microbiome of their babies is right in the middle. But when they do another type of analysis, they could see that the vaginal microbiome from the mares, it was responsible for donating a lot of species. In this case, over 22 of the species present or genera present in their babies, it came from the birth canal, while fecal microbiota was also very important in that first microbiome that those foals were acquiring. So this is very interesting because when we go to ruminants, because they have this behavior of licking the mouth of the calves right after they are born, and that has been shown in calves and also in small ruminants, their microbiome is more similar to the the oral microbiome of the cows, which is the bacteria first, or they overtake the bacteria that were acquired in the birth canal. So, but we don't see that in foals. And why is that important in horses or in general? There is a lot of debate about it doesn't matter. Yes, it's very important, the first bacteria arriving. So some of you might have heard about the hygiene hypothesis that tells that kids growing in a farm, they don't get allergies, they don't get asthma. And kids from the city, that grow in a bubble, very clean, with a lot of hygiene, they are more likely to develop those kind of disease, which I'm not going to go into details or debates here, controversies, but there is this theory, and many studies have shown that there is a different type of immune regulations if you are exposed to a high diversity microbiome when you are born compared to if you are very clean. So in horses, We don't have anything in foals, but what we have now is a couple of evidences that in adult horses, a few species might be related to the immune regulation of the systemic immune system. Okay, so those are intestinal species that if they are present in high abundance, they have a different expression of some genes related to the immune system. And these other reports show that if you remove the parasites from the GI tract of horses, you kind of stimulate some inflammatory response, which agrees with the hygiene hypothesis that combines that the high diversity bacteria, but also parasites, they are necessary to regulate the immune system, because we kind of evolved in nature having them, and if you take them out, you develop some chronic inflammation. So this, I think, the future will tell us, but what we can extrapolate from that is that what we do with horses nowadays is to change their microbiome, the microbiome that has been selected in nature for them to have. As you know, horses have been selected to graze all day and to stay in the pasture all the time. But what we do is to take them out of the pasture, put them inside of the stall. We feed them with a completely different diet, like I showed. We change the microbiome because we give them high energy, soluble carbohydrates, and we exercise them. We travel with them for competition, and also we use antibiotics like water, right? So we are just changing the normal microbiota of those animals. But my question is, what's the consequence? Because even in humans, the fecal microbiome, it contributes to the composition of the vaginal microbiome. But now let's just think a little bit in horses, because of the anatomy, that the composition of the fecal microbiome, it might even further contribute to the composition of the vaginal microbiome. And this might have an impact in foals, which we don't know yet, but that's a theory we have. So the consequences of this altered microbiome in colonizing foals, there are no studies yet showing, but I'm just giving here some food for thought for you, because those are some questions we have, and especially like talking to specialists on equine asthma, they also agree that this could be something interesting to investigate. Okay, so are those horses having asthma more likely to have because they have an autoimmune system that is hyperreactive, just like we saw in humans, because they were poorly colonized? That we don't know yet, but in this study we did with Mathilde Leclerc, what we could see is that they do respond differently when you change asthmatic horses from a normal diet, when you change to a moldy hay and you induce the exacerbation and the clinical signs of asthma. Their intestinal microbiome responds differently from healthy horses. So there is something going on there at the lung level, and of course there is a lot of systemic inflammation going on those animals, and this might also affect the intestinal microbiome, which is difficult to establish a cause-consequence here, but we are starting to get pieces for the puzzle to have a better idea about why this is happening. All right, so I hope I'm not going too fast to Vivian, but I'll try to slow down a bit. But here, what we're gonna see now, it's the composition of the foal over time. So longitudinal changes that happen, just like in humans, when we are born, foals, they are colonized by every single bacteria they get in contact. Okay, everybody wants to be there. Like I said, it's a good environment, it's full of food, and initially there are going to be a lot of bacteria that we call facultative anaerobic. But in this study, what we did was to compare the microbiome of foals from the day they were born until they become 9 months old. and then we compare it to the feces of their mares, the adult horses. The first thing we can see here is that 1-day-old foals, they have a very specific microbiome. Like I said, everybody's trying to establish there. And those animals are just nursing colostrum. They are under heavy influence of IgGs, IgAs, and over the first month of life, we saw a big shift in the composition of their bacteria, right? So here they are still under the impact of milk, which is the main feed source for those animals. But as soon as they start to eat a higher amount of forage, that microbiome starts to change. And as you can see here, it changes in a predictable way until they become 9 months old, which is more similar to the feces of the adult horses that we can see here in black. That's very interesting because if you give me a fecal sample from a horse, I can tell you the age of that animal without even looking at the horse. So based on the microbiome composition, I can tell you this is a 1 day, this is a 30 days old, or this is 2 months or adult horse, just based on the type of bacteria that is present there. All right, so in this other study, what they did was something similar to what I did. That first study I showed was result of my PhD that I did in Guelph with Scott Weiss. But now here they are looking into more specific dates. Okay, so day 1, day 7, 28, and 60. And when they are 60, and also they checked right after weaning when they were about 6 months old. So what we can see here is an agreement with the results we had. Newborns, they have a very specific microbiome here in red, and that shifts completely after 1 week of age, and that changes even further during after 4 weeks. Okay, and that keeps changing in a predictable way that is coming more and more similar to the adult horses. And in this case, they were even able to see a change right after weaning, just like we saw too, an increase in certain types of cellulolytic bacteria that it is observed after weaning. Again, another study showing the same pattern, just shifts from when they are born. So I guess at this point, we already know it's very well established that the microbiome of those foals will change over time. But in this study, they did also very specific time points. So here, it was the meconium, just right after those foals were born. And here, as you can see, a big difference from day 0, or right after they're born, to the first feces during the first day of life. So in a matter of 24 hours, and also have to have caution to interpret this, because this can be just contamination from the environment, as the meconium is supposed to be sterile. And then you have a big shift over the first day, which keeps changing over the first week. And now what we are trying to understand is that those shifts, what are the factors impacting these changes in the composition? Because at that age, that's when foals, they start to eat the mother's feces with coprophagia. And also that's the phase where we have the foal heat diarrhea. So I think we know now, and those studies prove to us that the foal heat diarrhea, it's associated with a big change in maturation of the intestinal microbiota of those foals between 7 and 10 days of age. And that's probably the cause of this hyperosmotic diarrhea that we observe in those animals. And also, which is still coming into the direction of the mother's microbiome. Okay, so there are not many studies evaluating the microbiome in foals with in sick foals. Okay, we do have a few now in adult horses, but this study from Angie Schuster, she showed that foals with diarrhea, they have a completely different composition from the controls in the same farms. As you can see here, the blue dots are the foals with diarrhea, and we have different microbiome composition compared to controls. But also what's interesting and consistent with the findings in adult horses and in humans is that foals with diarrhea, they have a decrease in the diversity. That means they have less species of bacteria there. The inflammation, the diarrhea, everything is causing a decrease or killing a lot of species, and that's associated with a decrease in diversity, which is most of the time not a good thing to have. Okay, we want a higher diversity. And here, just to exemplify studies that we know that in adult horses, we have a larger sample set that's well established that diarrhea can cause dysbiosis, which is basically a big shift on the intestinal microbiome of those animals. Okay, so that was not too hard, when they invite me. I said, well, I can talk about the full microbiome because it's an easy topic. There are not many studies. That's what we have so far on the intestinal microbiome and the development of foals. But now what is interesting, what really interested us is how we can change this microbiome, how we can treat the dysbiosis that we see in foals with diarrhea, and how we can use this data to improve health. And I think when we talk about microbiota manipulation, the first thing comes in mind is probiotics, right? So I have a full talk just about probiotics. And what I want to say is that this is quite controversial. In humans, in horses, in foals, for every study that show a beneficial impact of probiotics, we have another study or two other showing no impact of those supplements on the microbiome. And I'm not saying it doesn't work. I'm just saying that we need more studies and better products, like we're gonna talk in a bit. But in this study, again, Angie, she showed that the use of probiotics, it changes very little or almost nothing the microbiota of foals compared to control groups. They could see a big change over time, like we saw that it's normal and physiological, but no difference between the groups treated with the probiotics or not. And that's not a fair battle, okay? Because the probiotics, they have first to survive the GI tract. They're gonna go to the stomach, they're gonna go through the GI to the small intestine and face digestion, and they have to travel and reach the cecum, and then after they have to, if they come out alive, they have to establish colonization in the colon, which might be quite difficult, okay? So we have to keep in mind that probiotics are normally safe. They might work. I normally prescribe because also the horse owners, they expect you to prescribe something to treat their horses. So do not hesitate to use it, but just keep in mind that they might have difficulties to work. For example, let's look at this, the composition of one commercial product market for horses. And you're gonna see that, they have four species of bacteria and one yeast. But when we did this study, what we saw is that, for example, lactobacillus, which is the main strain we use, they're very, very abundant in the stomach and the duodenum of horses. But then the abundance in the distal gut, it just decreases to, almost less than 1%, almost zero. I'm not saying they don't work. I'm not saying they're not important there, but what I normally try to say is that maybe there are other species that are more adapted to the distal gut, like some species of Clostridia or some species of Ruminococcus that are present in the colon at high abundances, but we normally don't think about using them as probiotics. This is what a group from Cornell did for calves, they saw that, for example, Faecalibacterium was very abundant in the feces of calves, and they started using that as a probiotic, and they were able to show clinical improvement in diarrhea or decreased incidence after the use of those probiotics. And here, just to give an example, a product that or an approach, I should say, that they use in humans. This is a multi-strain, like they call an ecosystem. They use over 30 species of bacteria, and some of them species that we never think about using as probiotics, like E. coli or species of Clostridia that normally we think there are pathogens, but many of them can be commensal, and why not used to reestablish the ecosystem facing diarrhea or dysbiosis. This is the approach we are developing in our lab. So we have an aerobic chamber, and we are doing culture of those difficult to grow bacteria. And maybe in the future, we want also to develop those ecosystems or complex probiotics to be used in horses, which it might have a huge use in the future. For example, when they evaluated the microbiome of high performance in endurance humans, they found one species of bacteria that was present in high abundance on those high performance athletes, which was Veillonella. And then when they gave that to mice, they saw that the group receiving the probiotic, they had a 13% better performance than the control group. So now the hypothesis is that this bacteria, it's able to metabolize lactate and transform that into propionate, and they think that can improve the muscle metabolism, and this can be associated with high performance. And why not in the future we might be using those type of selective bacteria to be present in high abundance in the guts of athlete horses to improve performance. And there is this first study from Nuria Ma. She's a Spanish researcher working in France, that in which she kind of suggests that and shows that high performance endurance horses, they might have some specific trends in their intestinal microbiome. And if you are interested, you can go over this publication to learn a little bit in more details about how this microbiome is able to metabolize this lactate and improve performance. Okay, so probiotics, as you know, are live bacteria. And then we have prebiotics, which are fiber that are used by the bacteria, but they are not digested by the host. There are not many studies in horses, in foals, using prebiotics. Prebiotics, you have to be cautious because they can actually kill a horse if you give it too much prebiotic or oligosaccharides. That's a model to induce laminitis, right? So you can feed a lot of the wrong bacteria, such as Streptococcus, for example, decrease the pH of the cecum, killing a lot of bacteria and causing laminitis. So prebiotics are very powerful, and we have to be careful when we prescribe that. There was one study that I was also a reviewer, that gave a prebiotic to foals, actually to the mares, and then after to the foals. And what they saw is that when foals were a bit older, at the beginning there was no effect on the microbiome, but then later they started to have different changes, and they saw an increase in the Akkermansia, which is a bacteria that is normally associated with health. But also what they don't mention in the title is that they had the increases in other bacteria that are not associated with health. So my opinion about that is it might have a very strong impact, and we have to be a little bit careful, and we need more research. And then when I was preparing this talk, I came across a recent publication from last year suggesting that fecal microbiota transplantation might be a good thing to manipulate and to correct dysbiosis in foals. So there are no studies evaluating FMT or fecal microbiota transplantation in foals. This is the first important thing to say. The second one is that even in adult horses, it is still controversial. Some studies show changes in the microbiome and other studies show no changes. So we are doing research trying to understand better that, but at this point, I would not recommend doing that in foals because there are many controversies like donor selection, transmission of pathogens, even the beneficial impact of the procedure, okay? So this is something to come in the future. Now, the other future perspective that I'd like to share with you is the concept of precision medicine, which tells us each individual is unique and therefore they have to be treated differently. Now, this publication from last year also, what it shows us is that they were comparing the microbiome from 2 farms of horses, and they showed that the environment is very important also. Those horses were receiving the same diet, same management, but just by being in 2 different farms, they had a different microbiome. And this another study studying the skin microbiota of horses showed the same thing. Depending on the farm the horses they have a completely different microbiome. The same thing in calves. This study shows calves from 2 different farms, different microbiome profiles. We don't have data in foals, unfortunately. And this is also microbiome of milk from 2 different dairy farms. So I guess at this point, there is no doubt that the farm, it's an important factor. The environment is an important factor for the microbiome. And what they are doing in calves now, and we have a similar study going on in our lab, is to starting treating farms like an individual. Each farm may have a completely distinct microbiome in their animals. So sometimes one farm might be a decrease in this certain bacteria, for example, Prevotella, and you have to develop a specific probiotic to treat the dysbiosis of the farm, not only of those foals, but also to correct the microbiome from the environment. So my prediction is that in the future, we're going to have a kind of a easy and quick test to do in which we can go to a horse barn and say, okay, this environment, it lacks this type of Clostridia, so let's formulate a recipe of a probiotic here to correct the dysbiosis of your foals. Right. In this other study from Nuriama, what she showed, she was trying to investigate the impact of 2 different methods of weaning in foals. So one group was abrupt weaning, just take the foal away from the mare, and the other group was progressive separation, okay? And she was evaluating the impact on many parameters, stress, microbiome. And what was very interesting that she saw was that regardless the method of separation, what she saw is that there were 2 types of microbiomes on those foals, actually 3, but she further studied 2, which they call enterotype 1 and enterotype 2. And what they saw is that depending on the type of bacteria those foals had, they had a different phenotype. One group had a low diversity microbiome, high levels of cortisol, low production of volatile fatty acids, and high loads of parasites. Now, the other group, we had completely the opposite phenotype. So we could now associate a good microbiome and a bad microbiome. Is this a cause or a consequence of those phenotypes? We don't know. The study is not proving right now, but that starts to give us an idea about which kind of bacteria we want in the gut of our foals. Now, this is the last future perspective that I want to give it to you, is that what we call the gut-brain axis. And this is huge in humans because the innervation of the intestine is as big as the spinal cord. So we call that the enteric nervous system. And the bacteria in the gut, this has been proven that it's constantly communicating with the brain by stimulating this enteric nervous system, but also by producing neurotransmitters, for example, serotonin, and this can go directly to the brain. That's why sometimes we refer to the intestinal microbiome as the second brain. And there are a couple of studies in horses that suggest that, but again, we don't know about that in foals. But something interesting to think about in foals is that what they see, they also show, is the modulation of the woman microbiome. For example, women treated with antibiotics, it kills their intestinal microbiome, and that can affect the neurodevelopment of their babies. And this might be something interesting also to think about in horses, because the amount of mares treated with antibiotics before and after foaling, it's just huge. All right, so to finish, I would like to give you some conclusions that the intestinal microbiome of foals, it changes from the day they are born until they become adults. And also that the management we currently apply to equine production, it's completely different from what those animals have been selected in nature to harbor, but we are not sure about the consequences that might have in their health. And finally, that in the future we might be using microbiome manipulation to treat disease, to correct dysbiosis, and maybe to improve performance in athlete animals. And with that, I would like to again thank you. And I'm not sure if you are answering questions now or later, but I'll be available to answer your questions. Thank you very much.